Thank you very much for the invite. So I wanted to first introduce you to our service center, Taylor. So we are located in the Texas Medical Center. In case you don't know, this is the largest medical center in the world. There's 10 million patients that are seen yearly, 180,000 annual surgeries, 750,000 ER visits per year, 9,200 patient beds. This is home to the largest children's hospital in the world and the largest cancer center in the world. So these are a lot of patients that could be susceptible to these antibiotic resistant infections. And so that's what we're dealing with right now. We're in the antibiotic resistance pandemic. According to data in the year 2050, 10 million people may die due to antimicrobial resistance. And in the US, particularly, the numbers are very high. So 150,000 to 160,000 people die per year due to multi drug resistant bacteria. And so we really need to develop phage therapeutics or alternative therapeutics for these patients, but we really need it right now. And so that's why we started Taylor. And so what we do is we actually find phages that are effective against multi-drug resistant pathogens in patients in our partnering institutions, either locally and also throughout the US. So here in the US, phage therapy is not approved for clinical use. So each case is a case-by-case basis where we have to apply for this expanded use and get approval so that the patients can be treated. And so what typically happens is that a clinician contacts us that has a patient with a drug-resistant infection. We look through our library to find phages that are lytic against it. If we can't find phages that are effective, then we go out into the environment and find phages. We then further characterize these phages, sequencing them, determining them to be suitable for clinical use. And then we purify them, removing any undesirable elements and ensure that they're sterile for clinical use. We then do further characterization, and then we send the phage cocktail to the patient so that when the clinician applies and gets approval for this expanded use, they then treat the patient with the phage. So because we are an academic institution, our phage therapy program is guided by the basic science. So what does that mean? So I'm going to take you through one of our clinical cases and how we developed a phage cocktail for this patient. And so this is work by Austin Terwilliger. And so this is one of our first patients that we treated, a 56-year-old with a chronic UTI and prostatitis. He has been sick for 3 years on continual antibiotic therapy. We gave them this 4-phage cocktail, a course of 2 weeks via IV, and the patient cleared the infection and has remained asymptomatic for the past year. So the phage cocktail was effective at treating this patient. So how did we actually develop this phage cocktail? Well, for that, I'm going to take you through some of the science and specifically about this pathogen that I've been studying for the past few years. It's an E. coli. It's called extraintestinal pathogenic E. coli. This is the #1 cause of UTI. If you have a UTI, it is likely caused by these pathogens. They're also the #1 cause of adult bacteremia, gram-negative, #2 cause of neonatal meningitis. So these types of severe infections can lead to 80,000 deaths per year. So we wanted to find phages that could treat these multi-drug resistant pathogens. And so what we did is we went out to the environment and we found these phages and we tested them in animal model systems. And so this is one example of an animal model system where we take mice and we inject them. This is intraperitoneal with a 10 to the 8th dose of one of our clinical isolates. An hour later, we then inject the mice with a 10 to the 9th PFU dose of one of our purified phages. This is HP3. After an overnight infection, we then look at the health of the animals and then we euthanize them to plate their organs specifically for this E. coli. So this is a disease severity chart where 0 indicates that the mice were not sick and a 4 indicates that the mice were very sick or moribund. And you can see that in the mice that received the phage, they got or did not get the phage, they were very sick and moribund, and those that did receive the phage actually did very well. When we plate the organs specifically for this E. coli, and this is in log CFU per gram of organ tissue, we see about a 3 to 4 log reduction in bacterial burden. So we could use this phage that we got from the environment to treat the severe bacteremia and to reduce disease severity. But these E. coli actually colonize us. So we are asymptomatically colonized by this bacteria. They're opportunistic. And so we really wanted to see if we could target these bacteria, remove them in order to prevent infection. And so what we did is we took another mouse model. So in this model, we give an oral dose of the E. coli. We can colonize the animal for a week and for months on out. A day into colonization, we then started to treat this animal with the same phage that worked effectively in the bacteremia model, a 10 to the ninth PFU dose given orally every day. Another group of animals actually received the phage in the water, and then a third group of animals received an antibiotic in the water. So this particular E. coli was susceptible to an antibiotic, so we used that as a control. So looking at colonization data, this is plated fecal pellets and log CFU per gram of fecal pellets. And you can see that in the groups that either received phage via gavage or received phage in the water or did not receive phage, we see no significant difference between the groups. But in the group that received the antibiotic here in the pink, you see a precipitous drop in E. coli, and no detectable CFUs throughout. At the end of the experiment, when you plate the small intestine and the large intestine for phage, you see in the animals that did receive phage, there are phage present in the intestine. And then when you plate specifically for these E. coli again, you see no significant difference between the groups, except in the group that received the antibiotic again. So this told us that the intestinal tract may actually be a prohibitive environment for phage infection. And so we took this out further using an ex vivo system, and we were able to determine that the inhibitor was actually mucins. So mucins are glycoproteins that make up your mucus layer. And our ex vivo assay, we take healthy animals and we euthanize them and we remove their cecum and we use the cecal contents as growth media. So this is just cecal contents with saline. We perform these 4.5-hour growth assays. And to ultimately determine if mucin was the inhibitor, we actually took the cecal media and we centrifuged it at a high speed to pellet these large glycoproteins. And then we resuspended that and used that as growth media. And we used the supernatant as growth media. So that was our soluble media. And then the growth media with the large glycoproteins was our insoluble media. So this is after a 4.5-hour growth assay. And you can see in LB that the E. coli grow very well. When you add this HP3 phage, it works really effectively. Now, when you take the cecal media instead of the LB media, you see no significant difference between the groups. And when you use this insoluble media, that is these pelleted glycoproteins that were resuspended, you see some inhibition again. And when you use the soluble media, the supernatant, you see that the phage start to kill better. Now, when you add an N-acetylcysteine or NAC, it's a mucolytic drug, to the insoluble media, you see suddenly that the phage start to kill better. And when you add purified mucin to your soluble media, you see this inhibition again. So this told us that mucins were inhibiting phage infection. And so knowing this, we actually screened our entire phage library that were lytic against this bacterial strain in LB plus 1.5% mucin to find phages that were effective under these conditions. So we were able to find a phage, this is ES17, that could kill under these conditions. Now, if you were to do this assay in LB only, in LB culture, you could see that there were several phages in our library that were really effective against it. And really, ES17 isn't even the most effective in LB compared to many of those phages. So ES17 is pictured here. You can see it looks a lot different from our previous HP3 phage. It's a potovirus. It has this elongated capsid. So we wanted to try this phage again in our mouse model, our colonization model, to see if it was effective. And so again, we give an oral dose of our E. coli to our mice. We track it over the course of the week. We then gave an oral daily dose of our HP3 phage that wasn't effective last time, or we gave them our ES17 phage. But this time we tried to give them 10 times as much more phage than we previously did. And so this is because we thought, well, what if we just gave the mice more phage? Could we overcome this inhibition effect? So after about a week, we plate the organs for the E. coli, and we see here that with HP3, it's actually very effective in the small intestine, now with 10 times more phage. ES17 is also very effective. There was only one mouse that was colonized by this E. coli. But when you look at the large intestine, you can see that in the HP3 group, there were still some mice that were colonized by this E. coli. And when you look at ES17, you see that it's almost just as effective in the large intestine as it is in the small intestine. And so we later learned that it's because this phage is actually utilizing mucins as a receptor to better infect these bacteria that are coated with mucin in your intestine. So this kind of led us to believe that the environment really matters. So not only does the phage have to be lytic against the pathogen, but the phage actually has to work within the environment that you're going to put it in. And so we put this into consideration when we develop these cocktails. So we need phages that are not only tailored against the pathogen, but they're tailored in the environment. A second part of this is resistance. So another really important thing to consider is how the bacteria are going to resist the phage. And so this is work that Keiko Salazar has done in our lab. She has characterized the X-spec resistome. So she has taken different clinical isolates of this E. coli and used our phage library to characterize genetically the different resistant mechanisms of these bacteria, as well as phenotypically. But she went a step beyond that and developed phages that were actually lytic against these resistors. And so how she did that is by incubating bacteria and phage, so phage with the bacteria that's susceptible, and then taking that phage and incubating it with bacteria that's resistant. And it's just this continuous chemostat system that allows you to easily isolate these adapted phages. And so knowing how easy this is, and so she was actually able to use a phage that she evolved to work against these culture-derived resistors that are pictured here. So they were culture-derived resistors as well as animal-derived resistors. So knowing that we could actually do this quite easily, we have integrated this into our program too. So phages in our cocktails should anticipate resistance. We also need to consider the actual clinical condition. So this is work from Belkis in our lab, as she has characterized cocktails that are broadly lytic, effective against 80% of all of the UTI isolates that we get from patients at a local VA hospital. But also she tests them under biofilm forming conditions. So this is a biofilm forming assay. So this is very important because these patients, they have catheters and they likely have biofilms on their catheters, and that's how they're getting these chronic UTIs. And so that's something that we also consider when we're building these cocktails, that they are unique for the clinical condition. And finally, we know that phage and antibiotics may actually work well together and synergize. And so this is work from Carmen in our lab, and she has actually developed an assay, it's called cyanography, where you incubate antibiotics and phages together, and you can easily determine whether these phages are actually synergizing with the antibiotics, antagonizing them, or maybe there is no effect or an additive effect. And so what she has found that this synergy is actually dependent not only on the antibiotic, but also the phage and sometimes even the pathogen. So this is really important when we're actually developing these cocktails for patients. So we take every phage cocktail that we develop and we test them using this assay and the antibiotic that the patient will be on throughout treatment. So phages should work effectively with drug combinations. And so going back to this clinical case that was effective, we used the phage cocktail, we could treat the patient with a chronic UTI. Again, this HP3 phage was effective in our animal model systems. HP3.1 was effective at targeting the resistors. ES17 is a mucosal targeting phage, perfect for this environment where these bacteria are present. And this ES19 phage was actually effective in these biofilm forming assays that Belkis had characterized. And then finally, we found that this phage cocktail had an additive effect with the antibiotic that the patient was going to be on. And so that is how the basic science can actually make the phage therapy program better. So really quickly, how are we accelerating this? And so what we're doing is actually using technology to better adapt these phages quickly. So we've turned this chemostat into an automated system and integrated this into a digital format so that we can easily kind of monitor these cultures and determine whether the phages have overcome these resistors, and we can isolate them quite quickly. We've actually quickly turned our purification system into a 2-hour process as opposed to the typical 5 to 7 day process of cesium chloride purification. And so this is, in summary, what we're trying to do with our focus, of course, is the patients in the middle, but also the basic science that we're getting from this patient data are helping us build better cocktails to treat the patients. And the technology that we're developing is helping us accelerate this process. And then a third leg of this is, of course, the outreach, reaching out to clinicians, regulatory agencies, other science institutions to kind of help patients and inform them of what we're doing here. So I just wanted to thank everybody and our partner institutions, and we're happy to partner with any other scientists, clinicians, academics, or companies out there. So thank you.